where I'm sort of still a visiting fellow at the LSE, where a lot of this work actually was developed. So I should give them some like hosting credit as well. And thank you all for inviting me here. I'm really excited to be talking to many of you over the next 3 days, in fact. And I'm a top believer. I'm also wearing, coming from Scotland, a shirt outside in wintertime. That's quite an amazing experience. And with this seminar today, I hope to sort of outline a little bit of where a lot of my mind is at, and the main reason, I guess, why Zach invited me here, because we are working on the problem of reproducibility, and part of the collaboration we'll have entails sort of introducing this idea of complexity within our understanding of reproducibility. Because what I'll try indeed to argue first in this talk is that I see clear evidence that this is an element that is entirely missing in how we understand this problem of reproducibility. By the way, before I go there, in fact, if I say reproducibility crisis, or even just how many of you have heard that science is in a reproducibility crisis? Raise your hands. Okay. Okay, that's almost everyone except you who weren't quite sure. I think you went along with the crowd. That's an excellent point, because I think too many people in this field do exactly what you did and go along with the crowd, and I'll show you what I mean. But basically, this is where we start from. Okay, there's this perception and the idea of reproducibility. What is that? Is the idea that when you replicate a study, the results should look pretty much the same. And so you have high reproducibility when you have that. Supposedly that is indeed sort of how science works in terms of producing findings that are reliable and other people can build upon. But I'm going to argue actually it's more complicated than that because of complexity, if you like. And with that in mind, having hopefully convinced you, or at least illustrated to you where I come from with seeing a problem with the way we tackle this idea, I'll illustrate what I've been working for a while and what I think is a possible solution, which has some evidence in support of it, and that's what I'm going to show you in the last part of the talk. Although, again, much of my spirit remains that I'm just going to offer this as an example of how I would see this problem of reproducibility and generally our approach to doing science of science, i.e. meta-science, to play out in a way that is more true to the problem and the science itself. This. So what do I mean by complexity, first of all? Well, I think about it in the very sort of technical, indeed physical sense of something that applies to a system, and a system is anything that is composed of interacting parts, if you like. And basically, a system is complex in a very, again, at least fundamental and basic sense. The more it has many parts that are diverse, like different from each other, and that interact in diverse ways. And this is equivalent to saying that the system is also going to take longer to describe, and that's typically the way you would measure the complexity of a system. And it also is difficult to predict in its behavior. So the properties of a system, in a sense, are less controllable, less predictable. And so what you see here, if you imagine this being a scale of complexity from left to right, and these being schematic examples of systems, and it's no coincidence that I represent them as graphs, i.e. collections of nodes and relations, because I indeed strongly believe, and you'll see why and how, that basically graphs all the language by which you can understand systems and knowledge in general. So this is actually key. But here, just as a sort of schematic example, pictorial representation of a system, you have the elements being these circles as the, whatever the system is about in terms of the parts, and the arrows are the interactions between the parts. And basically from left to right, in the upper row, we increase complexity because we're increasing the number of elements and the diversity of them. Perhaps less obviously, a system such as the one on the bottom left would be less complex measurement-wise than a system in the middle, simply because even though it has seemingly many interactions, for example, many more than this system here, it is perfectly symmetrical. It's easy to describe. You know, you give two numbers. In fact, you give one number, really, the number of elements, and then, you know, with a couple of instructions, you could reconstruct the entire system. So this is actually relatively simple, whereas this is more idiosyncratic. You have to describe the relations in specific ways, is going to be technically more complex, and all the more, of course, if again you start to consider more diversity of parts and interaction. By the way, so we had to convert the presentation to a PDF. So for one thing, I'll keep clicking on the wrong button to change it for at least half of the presentation. And the other part that saddens me a little is that some of the sort of punchlines or things I would have asked you in advance of showing you then the answer will be given away. And so if I remember what's coming next, I'll try to ask the right questions before. Otherwise, such as in this case, you basically probably infer the answer even before I ask the question, because although luckily this I asked already, is there a reproducibility crisis? Well, again, lots of people say this, and effectively the field that by now people such as Zach and myself are in is entrenched in this idea that there is a fundamental problem with, for example, how well studies will replicate, i.e. reproduce. And not only that, but that largely this problem is driven by problems with research practice, with how essentially researchers either sort of engage in poor reporting and poor sort of approaches to research in general, or indeed even worse, they engage of what are known as questionable research practices, meaning that whether intentionally or not, but they engage in forms of behavior that basically increase the likelihood that whatever they say is false, in a statistical sense and in an empirical sense as well. And I used to believe exactly the same thing. In fact, I've probably been instrumental at the beginning in promoting this idea based on evidence that I was collecting at the beginning of my work in this field, which by now is over 10 years, in fact, more than that ago. But then once I started to see results such as these that came out from studies that took samples of studies from the literature and systematically tried to replicate them, and they tried to measure what the results were, I started to completely change my mind. And the reason, the main reason that made me change my mind is in the circles that show the upper estimate that, according to these studies, who were purportedly measuring the level of crisis in reproducibility, the results suggested reproducibility would be like. But there's something to be said about the fact that measuring reproducibility in this way is already probably, in fact, according to me, certainly wrong. It's wrong to think of a research result as a binary that is either true or false or replicable or not. In reality, it's a matter of degrees. But nonetheless, even sticking to the standards that these studies set for themselves to establish how reproducible research is, you see that basically in all cases, at least the results were at least compatible with a reproducibility above 50%, and in many cases, well above that. And this is just a few examples, actually. Ever since I made this point, there's been several other studies that would get too long and a bit messy to add all on a slide. But the results are very typically similar to these. Okay, you get frequencies of 60, 70% of studies that, again, according to a fairly flawed and conservative, if you like, criterion, nonetheless, suggest that these studies reproduced. So why on earth would people say that there is a reproducibility crisis? Like, assuming, and this is where my point about going along with the crowd comes in, because assuming that people even know about this evidence, and I suspect that a lot of people in this space by now don't even look back at what evidence we have about reproducibility. They just assume that since everybody says there is a crisis, there has to be a crisis, and they'll raise their hands as well and go along with it. But again, I'm even assuming that they are aware of the evidence, then evidently the only reason why they can think that these estimates are insufficient, that reproducibility is too low, is because presumably they expected that in any of these studies, the frequency should actually have been 100% or maybe 95%, something in that order. That is obviously, I think, at least intuitively, unrealistic. I think most people would understand, at least again intuitively, that if they tried to take a study from the literature and repeat it, depending on the field you're in, sort of you might be more or less sure about the methods you're using and the kind of results you would get. But you wouldn't be entirely surprised if the first time you tried it, it didn't work, because you might have missed some key element, or the kind of phenomenon you're looking at is so idiosyncratic to a particular area. I'm thinking, for example, of psychology, such that doing it in a completely different, say, country or population wouldn't show the same results. And in other words, I think there would be all sorts of intuitive reasons why one wouldn't expect always to systematically and simply take a study from the literature and obtain exactly the same result. And this is where complexity comes in, right? Because again, at least on an intuitive level, I think what we all have in mind when we think along these lines is the fact that the world is more complex than the simplistic picture that one ideally depicts in science, and arguably sort of over depicts. Okay, maybe we're oversimplifying the problem, but again, rather than there being a crisis of reproducibility, I would argue there are the entirely wrong expectations. You don't need to take my word for it, because there's been a lot of people that since these studies came out, usually criticizing these studies, but not even necessarily, but have pointed out all sorts of factors that would lead you to expect a lower reproducibility than actually observed, and well, a lower reproducibility than this ideal 100%, regardless of the quality or honesty of the practices of the researchers who produced the original findings. This is a non-exhaustive list of factors that I'm reviewing as part of this project that Zach and I are involved in. Okay, and there's obviously too many different ideas for us to review, but for the most part, they revolve around, again, this notion that basically the phenomena you're studying are likely to vary, or likely essentially not to be fixed. At least you can't give this for granted. And there are plenty of fields, certainly psychology, which is where a lot of this evidence about reproducibility or otherwise comes from. Psychology is a field where phenomena vary a lot beyond the simple sort of sampling variance that you assume in statistics. And similarly, that the methods that you use might be more or less well codified, more or less mature, more or less advanced. And again, you assume that whatever is described is sort of a fixed measurement of what actually all you need to do basically to reproduce the same result, and that is a fallacy. There you go. What was that? Sure. Yes. I think some of the literature or not. So, and that's a good point, and thank you. I'll move on because I'm aware we're recording, so maybe I'm just going to guess. I don't know. You're the director of the recording, but perhaps as we follow along, it's better, and then we can keep. But it's a very good point, and thank you for that, because what do we mean by reproducibility, right? And so one of the things that we're doing with this project is, in fact, sort of clarifying this vocabulary, because it's all over the place, and people use the same words to mean rather different things. What I'm going to be talking about here is the empirical reproducibility, not of the kind that you have in mind. And you're quite right that the key element here to think about is that you're collecting new data. So the problem is that you take a study, but you collect new data, and you assess what the results look like. With the same data, the expectation could be higher, although again, the situation ultimately wouldn't be too different in terms of the kind of reasoning that I'm going to align in that case, in that case relative to this. But certainly, what we're talking about here is reproducibility as the result of a replication study, where a replication study repeats a study with a collection of new data. Excellent. And as I said, again, all sorts of reasons why intuitively that would lead to results that don't quite look like the original. And this is the elephant in the room that I'm referring to, because as shown in all those studies, for example, a few of them actually mention complexity explicitly, but they also cite my work, so they're probably sort of familiar with a certain way to look at things, but most of them don't. And yet, like in my understanding, exactly as the proverbial people in the dark room that they're all sensing an elephant, but no one sort of can see the whole animal. Basically, that's what they're touching upon. All these different reasons why, in terms of the kind of concepts one is using or measurements that one is applying to reproducibility or the expectations one makes, basically, one sees all sorts of reasons why these should be different from what we have at the moment. But the common underlying factor, I would argue, is this fact. At least it can be defined and conceptualized and measured in terms of the complexity of the phenomena and the methods underlying any particular scientific claim. The idea that this complexity varies across fields, it varies depending on the type of study. Now, some could argue that actually, well, that's unfair, because in reality, we do have statistical concepts that handle this kind of diversity, at least to some extent, and that would be what is often referred to as the random effects model. Where how many of you have done a meta-analysis? Raise your hands. How many of you have done any kind of statistics course to begin with? Okay, at least. Well, how many of you know what if I say a random effects model, a random intercept or random slope in a regression model? Do you know what I'm referring to? I think, okay, so I think that the raising of hands was fairly convincing in this case, and convinced. But basically, you know, the idea is that we will add to our model a sort of a second source of variance that says that, you know, possibly depending on setting or whatever other variable one might be considering that groups the data at a higher level, you have this extra element of randomness. That's fine. However, it's at best, very poor way to handle complexity, because all you're doing is imposing a new, fairly simplistic model about it. You're saying, okay, there's a normal distribution, a continuum distribution of this new variable, and this is just simply going to reduce effectively our statistical power or our confidence in the conclusions of our results. And that, again, is at the very least a very imperfect way to handle the problem. But in reality, again, I would argue that there's a lot more to this than simply conceiving of the randomness of errors beyond sampling errors. And there are many reasons that would make me say this. One of the most important that was very influential in my own thinking is the fact that actually key to the idea of complexity is that the kind of noise or unpredictability that you're confronting is not random. It's actually it's systematic. It comes from a structure, but it appears noise to you because of your ignorance. That's really the key idea. And these authors, in a paper that was actually preceded by a few years, this one, made this case very pointedly in biological research. In biological research, there's something called the reaction norms of reaction, which basically simply means that an organism who has a genotype and a physiology and expresses a phenotype of some kind, behavioral or, again, physiological, but basically is known that potentially will express that phenotype differently in different environments. In other words, the same kind of stimulus, for example, will induce a different result depending on some other third, fourth, or how those are many other variables. So that is known to occur, is in fact a known issue to take into account in laboratory research in those areas. And what these authors pointed out is that actually, if you think about it this way, again, you think of, for example, an environment that has this continuous reaction norm as a function that links the environment to then, whatever you're expressing in terms of the phenotype. But you don't know about this variable, you don't know that it exists. Then it creates a paradox where actually you have perfect reproducibility. You could have perfect reproducibility in those results, but you're running the same experiment in two different labs, the results look completely different, and you conclude that there is no reproducibility. And the more you try to standardize and increase sample size to make your studies more precise, the less reproducible they'll appear. But in reality, all you have is ignorance about a third modulating variable, effectively. So this is, again, it's a basic concept that potentially, is part of anybody's understanding of any phenomenon. And yet we go to reproducibility and we sort of ignore this and pretend that at best there is simply a bit more noise to take into account. All right, and so taking into account the problem of complexity is then what I would argue is really missing from meta-science, research on research, and certainly in how we're handling reproducibility, which to me is important because unlike perhaps other more basic areas of research where people produce interesting findings that might be correct or otherwise it has no consequence, in meta-research people are pushing for policies based on a certain understanding of what the problems are. So if that understanding is incorrect or at least heavily incomplete, then the policies we are imposing are just going to do more damage than good. And that's something that, as researchers, if anything, should concern us all. So I believe it is important to take complexity into account. And so how can we make, if you like, the metaphorical elephant emerge from the darkness? What I've been trying to do for a while is using this approach that I indicate as sort of K-theory, although rather I think it would be better defined as information compression, IC theory. I'll say at the end that I wonder if theory is actually a good word for what I'm proposing. Nonetheless, this is how I'll define it for now. But basically, it is a framework and a methodology that tries to incorporate this idea of complexity into an understanding of knowledge at a more abstract level than the usual nitty-gritty, rich statistical level that people sort of normally approach it in meta-science. The key idea revolves around this function, which is really sort of a metric of knowledge derived from, in a very simple way, from very basic first principles, with a few sort of key ideas around it that I'll stress. But basically, so this K function, this quantity K, is again, it's derived, okay? But once it was derived, what I realized, which is then, brought me into, again, developing these ideas more and more, was that actually it turns out to express pretty well what many people, and certainly myself, would understand scientific knowledge and ultimately knowledge essentially to be, which is a striving for consilience. So I think it's hard not to argue that basically what our ideal of a science does and it's striving to do is to explain or predict or control as many and as more diverse phenomena as it can using the simplest and fewest possible theories and methods. Ideally, again, like think of Newtonian physics and the kind of revolution it brought. It was a simple set of a few equations that all of a sudden were able to predict a huge range of phenomena and explain all sorts of things that people had observed before and weren't quite able to connect. So all of a sudden you can predict the position of planets decades in the future, predict the arrival of comets, and you can predict tidal waves in the ocean and so forth, all with the same system of principle. And this is really what is wonderful and useful about science. And again, it's not my idea. It's a very old philosophical idea, but like many good old ideas, I think it got lost over time. And again, without originally intending really to do that, it turns out that this function kind of captures this definition pretty well, because basically the orange bits, which I'll explain in a second what they are exactly, but capture what we're trying to get, okay, the greater diversity of phenomena. The green bit is essentially our ignorance, or the smaller that is, the better we are at explaining, predicting, or controlling. And the blue part is what we have to put in order to achieve that level of prediction or explanatory power, which in other words are our theories and our methodologies. And one of the tricks here, or the novelty, not as novel as it might seem, but novelty nonetheless, is that instead of these being ordinary sort of statistical parameters, these are all quantities of information. So the function h is Shannon's entropy, which now again I'm gonna just to know how to go on from here. Do you know what Shannon's entropy is? Okay, so it's a quantity of information, and I'll show you sort of in a sense essentially what information in a technical sense means. But basically, this is a measure of our ignorance. Or conversely, again, it's a measure of how much our ignorance can be reduced. And so, one of the reasons why this is an approach that is abstract, more abstract than ordinary research methods in research and research, is that we're no longer talking about the specific details of a statistical model or a parameter estimation or some phenomenon, where you're simply quantifying anything, is in terms of how much information we wish to have, which is the orange stuff, and how much we actually get, which is the green stuff, spending how much information in order to achieve that, and that's the blue stuff. And information, basically, in a technical sense, information, in a physical sense, if you like, or statistical sense as well, it's simply the logarithm of 1 over the probability of some event. So the information of that event is, so if you like, the negative logarithm of the probability of that event. And a logarithm base 2 gives that quantity in the number of bits. And what does a bit mean? Well, a very concrete and correct, in fact, way to think about any of these quantities, these h's or these that I'll show you throughout the presentation, is some kind of volume. These quantities cannot be negative. They're either 0 or positive. And the volume expresses the number of the minimum number of binary questions on average that you would need to guess some value. So, for example, if I have the stereotypical bag of differently colored balls, I could pick a ball and ask you to guess which number I picked. There's up to 15. Well, it's easy to show that basically the information that I would express in guessing or in having that answer, or equivalently, the amount of ignorance that I'm imposing on you by saying I've got a number and you don't know which number it is, it's log base 2 of 15. And that's the number of bits of information that that question entails. And Shannon's entropy, which is this h function that you saw before, it's simply the average of this quantity. So this is what information is. And Shannon's entropy is simply the average, the expected value, more accurately, of that quantity. The idea being that if I ask something that has a different probability distribution from the one in the example before, in other words, I ask you, for example, what color is the ball that I have, then you see that now I only have 3 possibilities, but with different probabilities, then this is the quantity I need. And it will always be smaller or at most equal to this quantity. Okay. But basically, again, just think these about volumes that express this quantity. That is a universal quantity that applies to any possible phenomena, as long as there are alternatives with some probability about them. And that is a measure of our ignorance, or again, of how much our ignorance is reduced. So that's what feeds into the k function. So all it's saying is that basically this k value is going to be larger in proportion to how much of the orange bits we have, either in terms of a sort of let's say a single question that could have had many possible answers that we get, or it could be many questions. It doesn't make any difference. And conversely, the quantity is going to be smaller the more either our ignorance, the green bit, is large, right? We still lack full information about what number the ball was, and or the larger the blue bits are, which is, the ball example doesn't quite work well here, but what kind of inputs and structure we have to put in place in order to get that answer. And again, it could be one, it could be many, doesn't make any difference. And so ultimately you get a value that approaches asymptotically one, the better the consilience, the more we're compressing information. That's why it's an information compression theory. It's zero when we have no knowledge, where our ignorance green is the same as the orange part. And it could even, in fact, go negative when the green, and under certain conditions, is larger than the orange. When we get a systematically wrong answer, then actually, in a sense, the number of bits of information that we need extra to just get back to zero, or let alone the correct answer, is larger than what we were originally trying to get. And so we have negative k, and that would be, again, sort of the worst condition of being genuinely wrong, not just ignorant. And I'll make you a quick example. You've all done statistics, so you'll know what a regression model is, right? Okay, so y equals alpha plus beta x. X is going to be our input, and plus there's some error. And so the entropy of y, not quite the value of y, which is what you care about in a regression model, but the entropy of y under the null model, when you have no explanatory power, that's what's going to give us the orange bits. And similarly, the entropy of the error is what corresponds to the green bit. And if you stop here, this quantity actually exists already in statistics as what is known as the pseudo R squared, used to measure certain models, such as the logistic model, for example, as a rough equivalent of the R squared statistic. And also, in fact, I discovered after having published these ideas, actually, that someone in sociology, for example, was suggesting that so just the orange and the green bits basically would be a measure of knowledge. So this is not actually, again, such an outlandish idea as it sounds, where there is more, something new that makes a difference here is in the blue part, is in the argument that, again, purely comes from a derivation of this quantity as a standardization of something that tells, well, actually, if you want to compute knowledge and how much knowledge you have, you need to include these factors here. Basically, the entropy of what in this case would be the independent variable x, and some measure of the complexity of the structure, and that's that d of tau there, again, expressed in terms of bits of information. And that is the methodological part here, where in a sort of a later development, like more recent development, basically, the idea is that we can use a graphical language, a particular way to describe any system in terms of a graph, as I hinted at the beginning, a graph that can have multiple levels, it sort of can be nested in each other, so arguably it can describe very complex structures. And this function d basically is going to churn out a number of bits of information to describe the graph. So that's how we plug in a measure of complexity in our measure. And again, this might seem a bit strange that you can describe anything in terms of a graph, but actually we do that already. So I'm sure you're familiar with statistical techniques by which we use graphs to describe things, such as structural equation modeling. And even informally, we might use, so we will use, in fact, often graphs to describe theories. And I think there's a very good cognitive reason for that. I do think that graphs work on us in a way that actually captures exactly what we mean when we say that we know something or otherwise, or that we think of any phenomenon as a set of essentially steps and connections between them. And plus, obviously, but even in methodologies, we use graphs to describe them. This is a startup that I think by now is being bought actually by some bigger group, but basically used to collect protocols from particular biomedical research, and they used a graphical approach to get people to describe these protocols. So all that we're saying is that basically there is in principle a way that we translate any system at this point that describes a knowledge claim in a graph, and we can derive the number of bits and plug that number of bits as well as the other sort of entropies that are easy to calculate in that function, and we get the quantity of knowledge of that knowledge claim. What this d function does, basically, I'm not going to show it because it's a bit long and pointless to show, but essentially what we're doing here is exactly what I was showing you before in terms of the example of information. It's a simple combinatorial calculus, such that given a graph, we calculate the set of all possible graphs that have the same structure in terms of the same number of elements, relations, and diversity of elements. and also with a simple mathematical introduction, a key detail that we add, basically, we can very simply extend that to the set of all subgraphs of that graph, which is actually very handy with these kind of calculations. So basically, again, there's nothing sort of magical about it. You literally get an integer which corresponds to the size of the set, which sort of very quickly becomes huge, and you're taking the logarithm of it as you would for any information. Basically, that's the complexity of the graph that describes whatever we're trying to describe. So this is the essence of the methodology. And a narrow way to understand what's going on here is that we are essentially penalizing a measure of effect size by the complexity of whatever underlies it. And we can do this literally in terms of statistical measures. So what you see here is on top is the R squared, the middle is Collins D, so classic standardized measure of effect size, and on the bottom is the chi-squared distribution. And you see that they all lie in a one-to-one correspondence with the value of k, which is on the y-axis. But depending on other stuff that we might vary about the k function, we get different values for k itself. So statistically, again, sort of this is sort of immediately showable. Now, the bolder claim, if you like, is that this is not just true of statistic. It's not just true in a sort of narrow sense, again, of regression models. It's something that we can, in principle, apply to any claim, any kind of structure. It doesn't need to be formalized statistically, as long as it's verbally described. And to the extent that it's verbally described in a clear way, then we can derive these quantities. And we have a metric on which to, I would argue, sort of operate in a proper science of science. Now, the reason why I don't call this just a methodology or a metric, but I proposed it at least in 2019 as a theory, is that it doesn't stop here, that in principle, just by sort of essentially algebraically manipulating this function, you get relations that answer metascientific questions. And you'll see an example in a second in terms of reproducibility. Basically, that's the idea. Now that you've defined what knowledge is, that function seems to have, at least theoretically speaking, all the elements that a knowledge claim does. And therefore, by working with that, you can get to understanding a problem about knowledge better and more accurately. So the reason that after these years I'm still blubbering about it is that I did get some supporting evidence that in some case, perhaps if I remember, I'll explain why, but I found compelling myself. And so I do think that there's probably something valid about this approach to begin with. But again, I'll stress now, and I'll try to remember to say it later, that I don't necessarily think this is the best way to go about it, but it's the only way I could think about. And I'll show you evidence particularly about reproducibility. Now, the first evidence I published already in 2019 is not the strongest, but it was encouraging because this approach seems to give you potentially new insights as to how one would understand the problem of reproducibility. So this is what K-theory would say reproducibility should be understood in terms of. So you have the K of a replication study, that's the K over R. And this is equal, with a very general sort of simple model, as the K of the original study times this 2 to the minus lambda D, where D, imagine being many divergences. So many things that will have been different, knowingly or not, in the replication compared to the original study, in the system, in whatever aspect of the system. It could be the phenomena, it could be the methods, it could be something else. And lambda is the impact simply that each of these Ds would have. Oops. Something missing. This sucks. Because actually here I had a graph that was supposed to be to appear. In any case, so I would have showed you this on a larger scale to show you what's going on. Now, if before you were falling asleep, now this is the death loop. But basically, what you see on the top there with these colors, the colors represent the size of the average lambda in a simple sort of simulation model. So the lambda, remember, is the impact of the divergence. So the larger the lambda is, the more drastically a divergence for, corresponding to that lambda will bring down the value of k, right? And in this simulation, basically from left to right, we simply imagine basically an average number of divergences. So it's very few here, and we increase them moving from left to right. And again, very simple approach and very simple simulation. But the point is that then what you have here is the distribution of results that we would observe, here expressed in terms of a correlation coefficient. So that on the top, you see when you have, for example, many divergences that are small as effects, basically you have a bell-looking shape, not quite, but basically is the kind of distribution that you would have, and some average reproducibility that is lower than the original. The original finding will be the dotted line here. Whereas you got the bottom, and perhaps in a situation where you have few divergences with a drastic effect on the result, then you end up in a completely bimodal distribution where either you have sort of a perfect reproducibility or things didn't work at all. So the idea is that these would be different kinds of fields with different properties. This could be something such as a hard physics experiment where everything is very well codified and already established. If you do it well, you get the result. If you don't, you get nothing. Whereas up there, these would be the more high complexity, low codification soft sciences. And within psychology, this is data from the largest reproducibility study done to date, which had 100 studies from the replicability from the psychology literature, and they had independent labs replicating them all. There is some evidence that this some support for this. The distribution of the blue of the effect sizes originally reported in these 100 studies, and this is the cortosis, in other words, the sort of the pointedness of the distribution. You see that it's exactly the same in the two curves, even though the ones above were studies that were rated by the replicators in that studies as being easy to replicate. Whereas these at the bottom were, again, rated by the replicators themselves as requiring higher expertise. In other words, if you do something wrong here, it's not going to work. And if you look at the distribution of the replication results, you do see actually sort of a difference here, where the bottom of basically essentially mostly clustered around 0, with perhaps a few that arguably worked, right? Whereas at the top, you have a more naturally looking distribution, which again would match what the simulation would have suggested would be observed. So this is kind of the approach that I would like to sort of expand more in terms of the project that we're here to discuss, in terms of how to try to predict reproducibility a little bit better, factoring somehow into account the properties of the system you're looking at. But there's been better evidence about this approach, which is really why I bang on about it still. Because actually, it turned out in a study that we did, we published a couple of years ago, and still just as a preprint, that K actually predicts how reproducibility is perceived. And this came from a collaboration with the Brazilian Reproducibility Initiative, which is one of these large initiatives. And they took 60 effects from the literature in biomedical research in Brazil, and they in particular focused on 3 particular methodologies. EPM, that basically is some behavioral experiments with mice, where the intervention typically is physiological, biomedical, but then you measure the behavior of the mouse. MTT is cell biology, so somewhere in between, if you like, on the scale of the phenomenological complexity, but not quite behavior methodological complexity. And PCR is sort of quantitative PCR, is the quantitative expression of genes. So something that in principle is hard, sort of easy to measure, but the methodology is quite complex and quite sensitive. Nevertheless, their ultimate goal is to replicate, have each of these effects replicated by 3 independent labs, and they're going to see how well these results replicate. But in addition to that, what they did was to ask a panel of experts to rate the likely reproducibility of these effects based on a summary of the methods and the results. And they got in touch with me because they saw now that I was doing this, proposing these measures, ways to measure the complexity of a methodology. And we started a collaboration in basically testing these ideas directly on these results. The data that they had already was the data on these predictions. So that's the first thing that we looked at. And so what I did before seeing the results was to measure basically the K of each of these studies, which is fairly straightforward to do. These are randomized studies, and I'll explain this so that you get a sense of how it works. But in practice, if you're conducting a randomized study and you have 50% treatments, 50% controls, it's a bag of balls with same number of balls, one of color, one of the other. That's one bit of information exactly. So they get a 1, very simple. And similarly, because you have only that one bit of information, the most that you can extract from the Y, the explanandum, that's also one bit of information. So in other words, you have maximum knowledge for that randomization where the whatever you're measuring is perfectly segregated between treatment and controls. When all the treatments are on one side and all the controls on the other, just as the balls would be perfectly sort of separated by color. And again, that's all the information you can extract from the experiment, and that again is 1 bit. And n is simply going to be the sample size, and it's the same, of course, for x and y. And then this is simply the average entropy of what you actually observed. How well segregated are treatments and controls along the lines? And so the last bit, again, and again, it's sort of the kind of the hardest one to do, is to measure the complexity of the protocols, of these methodologies that they did. And so what you have to do, this is the idea, you devise a coding scheme by which you organize a methodology in terms of a set of elements that, frankly, for the most part, these were the elements in the protocol already, barring a few differences. So you have basically a description of the subjects, S, and C, the experimental conditions, and CS would be, the result of the combination of these two. P is the procedure, and M is the measurement, basically. And within these, then you devise a scheme of what that looks like, again, in terms of a graph. And you can do this for as many levels as you like. Basically, until you're on these higher levels, you're creating a common structure to all of these experiments. And the bottom node is what guides you, is quantified in terms of the parsing of the text. Basically, at the bottom of each of these, of the nodes of these graphs in the bottom levels is the actual sort of parts of the sentences of this protocol that either are describing some parameter or describing the subjects or describing the procedure. This is in practice how the result looks like. So this is the graph. Okay, so this is a graph that describes the protocol of one of these experiments. You don't have the name, but it doesn't matter. And you can see that basically the stuff in the middle is the structure, that higher level structure that I told you before, and that is just a constant for all of these protocols. But you see at the bottom, well, each of these nodes is literally text. This is literally the text from the protocol. The idea being that, again, using a procedure that we try to make as systematic as possible, and we validated it amongst us, but I'm not going to say it's sort of easy to do or easy to automatize, but basically you parse the text in terms of these core concepts that are either describing each crucial passage of the procedure, or they are describing crucial details about the subjects, what age or weight the mice should have. Those would be 2 separate nodes, etc. Then you sort of plug this structure into the D function, and you get the number of bits. You have the other number of bits, and you simply quantify the K of the original study. And this is how well this correlates with the ratings that these experts, each expert of different methodologies, gave about the likely reproducibility of these results. So on the left-hand side, you have the replication probability elicited, and on the right is the relative effect size. And actually this, to me, is the only one for what I said before in terms of this false binary that we create about successful and not successful replication. I don't think this is actually a good measure at all. This is actually a way we should measure reproducibility or the perception of reproducibility. But either one correlates pretty well, and I'll show you in a second the numbers. But what I'd like to mention about this, which gave me some sort of hope about these methods, is that actually there was no exploration here at all. In fact, originally, by accident, I was given the wrong data. So I had the analysis, I had all the code presenting this completely flat null result until I realized there was actually something funny with the data itself. And so I got back, and Kleber, who's the one I was primarily in contact with, realized, oh yes, no, actually I was, sorry, an error in the coding. And so he sent me the correct file. And all I had to do was plug it in, and I got these results, which are fairly strong. Which again, like to me, this study wasn't pre-registered, but in my mind, this is as good as a pre-registration. Like, you know, this is a true effect that we're measuring here, whatever it means. I say they're strong because this is going to be a bit complex to explain, but basically the measure of the regression line that I showed you before in terms of the K factor is what you see here at the bottom. So this for the probability of replication and this for the relative effect size. And on top you have the indicator variables for the three methodologies. That, of course, is never going to change for any of the methods. We have to control for the different methods. And so, first of all, again, so this is the key result, and it's fairly strong. But interestingly, it's stronger than any other model that we tried afterwards to challenge this result. And in particular, what we investigated is the following. Okay, we get this great result with this, but is it because it's K? Like, is it because of this whole methodology that we said that we're measuring this way? Or would we get equal or better results by using ordinary methods? And this would mean, for example, simply taking the standardized mean difference, take the logarithm of it, instead of using the entropy and everything else. And then we had a rating given by these replicators of the subjective difficulty to replicate. So why not use that instead of d of tau? But again, you see, we plug these in, the effect size has some effect, but it's not quite as strong. And the methodology has no effect at all. So the difficulty has no effect at all. Then we tried, okay, let's take the ratio of these two. So this starts to look a little bit like a K, but it's a K not based on information quantities. It's based just on, again, normal quantities plus some structure that we give it in terms of a ratio. And again, there's a small effect here, but not nearly as strong as that of K. So we said, okay, but here we're log transforming in this way. So we produce the exact same measure, not based on entropy. But again, we get some effects, sort of like a modestly statistically significant effect for replication probability and not at all for relative effect size. And in any case, weaker effects than what we measure with K. So it would seem that there's something about all this framing that I described in terms of thinking about effects in terms of information, in terms of entropy, and thinking of knowledge in this way with this ratio where the complexity goes at the bottom somehow and penalizes in that particular way the entropy explained as a measure of effect size at the numerator, that essentially captures whatever these raters, these scientists were seeing in the replication results better than any metric that we have so far. But of course, this is just what replicators perceive about reproducibility. The real question, ultimately, which is why I still haven't published as a full study those results there, because when we submitted it, basically the reaction was, well, okay, but this is just the perceptions. Like, you should send this to a philosophy journal or a psychology journal, perhaps. But again, that's not really the point. You know, the point to me is that we know that these subjective predictions of replication correlate well with actual reproducibility. And so my guess would be, okay, that this is going to work, at least to some extent, in predicting actual reproducibility. And there is some evidence for that. So this is where, again, sort of a theoretical approach be derived from K. So we start again from the relation we showed at the top. Okay, a few passages, and basically you get to this relation where this becomes a metric of reproducibility. This is the ratio of basically the entropy explained in the replication versus the original. And on the other hand side, you have this relation. And this is a simple linear model that you can test with a simple linear regression again. And so, so instead of using K, basically, now we're focusing directly on the d of tau, the complexity of the replication protocol or the replication against system. And so we've derived theoretically a prediction, basically, from the starting point, which says that we would expect this particular relation between the complexity and the relative entropy explained. This is what it looks like on a sort of retrospective analysis of data from other reproducibility studies in social psychology and in cancer biology. And again, these are pretty strong effects. Now, the warning sign here is that these are exploratory results. So again, these are not as exciting to me as the ones that I showed you before, because I'm aware that sort of I tried different things. Many of them worked, so it looks pretty robust, but others didn't. And, God knows at this point, whether I should believe them or not. But certainly, there is a very strong relation here. More importantly, because, so first of all, this is the effect that you're looking at. And again, it's a very strong effect. So this measuring d of tau of the replication protocol basically strongly predicts the reproducibility that was actually measured in these studies. But all the more because in this case, again, one of the things that I tried, this wasn't the only way in which it worked, but here I simply, I didn't even use the whole structure, that graph that I did before. It was a simple graph where each node was one of the sentences in the protocol. So longer protocols, sort of longer graph, more d of tau, and basically this is the relation that you get. So if something like simple as this works everywhere, this is the simplest possible thing that you could apply to the entirety of the literature and get a prediction of replicability. But again, I'm not saying that's the case because there was a lot of exploration done. So, and in any case, it's not as simple as it sounds. Another thing that makes this result promising is that I wasn't just measuring the complexity. I'm controlling for the other two factors that naturally, based on ordinary, on current statistical thinking, you would expect to predict the reproducibility, which is the statistical significance of the original finding as the p-value. And this, I take the log because, you have to linearize the quantity. And effectively, the log of the p-value is a quantity of information, right? As we said before, any logarithm of a probability is a quantity of information. So this is effectively the surprisal of the original finding relative to the null model. And you see that indeed, at least in social psychology, it strongly correlates in and of itself with the replicability of the finding, as you should expect. But the effect we're measuring about complexity is independent of that. It's an add-on. And the other is sample size, again, for obvious statistical reasons. And in this case, actually, unlike the p-value, this seems to be relevant in cancer biology. But again, the effect of the complexity of the protocol is adding on to that, is that giving us more information to predict reproducibility. So the idea is that if we have this kind of knowledge, in principle, we can control, for example, for the complexity of a protocol and actually evaluate what is the level of reproducibility in a field, and therefore, perhaps at that point, make an estimation as to whether the results that we're getting are truly below what we should expect or otherwise. This is the idea, and this is how ultimately we would factor in complexity into meta-science. I'm not quite there yet, because obviously the climax of our collaboration with the Brazilian initiative is that we pre-registered the test of the actual replicability of these results to see if those same measures that worked so well on the subjective estimation and predictions basically will actually predict reproducibility. And these results should have been out in September. Since then, it's been one step on the other for perfectly good reasons. They want to make sure the data is correct. But I was hoping to show them here, and again, I had to be disappointed right up for Christmas that I couldn't. So, truth is that at the moment, I still have to see what happens with these results to know if this is truly a good approach to measuring actual reproducibility, or if it's just working so well for our subjective perception of reproducibility, which would be entailed by only having the previous result as valid. So I can't be sure about that, and this is really where I'll leave you. That just to summarize what I tried to say, there is an elephant in the room. That elephant is the elephant of complexity. It's a complexity elephant, and we basically we either don't see it or pretend that it doesn't exist, and that we can just talk about science and make models about science as if it's all the same thing. And I don't think that's really sort of appropriate, and it's actually quite damaging. And I think we do need to think about complexity in a more concrete and scientific way in this space. And I've tried to illustrate one way that I'm trying to do that, and I think it's quite promising. But I'm not going to say that it's actually sort of working, let alone that it's the best way to measure necessarily complexity. It has limitations that frustrate me, frankly. Because beyond a certain measure of complexity, I'm not sure how you can factor in, for example, the actual differences between different methods in terms of the different impact they might have, for example. And also, again, I call this a theory because I think it does something more than simply give us a metric. But I wonder if it's a genuine theory. I wonder, in fact, if K is actually a real number or not. At the beginning, I claimed that it was. I thought it was. So this is a genuine property of a system. I still don't know if that's the case. So this is where I leave you. Well, thank you very much for the attention, and I look forward to any questions and challenges, especially. Managed to kill every possible initiative. Oh, thank you.
So, in statistics, there's also
Yeah. Totally.
Yes, it would be design. Sample.
Yes. So, and that is actually discussed in the original paper. In fact, I'll tell you more. There are not just those information criteria, the archaic information criteria and the Bayesian information criteria, but there's also, as you probably know, an area of statistics called generally the minimum description length principle, which goes even further in actually becoming quite close to what I was suggesting here in terms of you're encoding the model in terms of a number of bits, and basically that sort of becomes your penalty. But we're doing something different in a number of ways. So first of all, those methods are entirely compatible in that anything that will minimize, in that case, the archaic information criteria, meaning, again, you're favoring a model that has a low AIC, that's how that works. The smallest possible log likelihood and smallest negative log likelihood, I should say, and smallest possible encoding on the model. Well, anything that will do will maximize K as well. But as I showed before, K has more things to it. So in a sense, it's expanding that logic. And the same applies to the MDL. So that sort of is the overarching point here, that that logic of minimum description length, or again, sort of an information criteria, at the very least, can and should, I would argue, be extended beyond the narrow applications of model selection. Now, the other aspect to that, which is something that originally, in fact, I wanted to show today, but then I've decided to focus only on reproducibility, is that there is very sort of debated, and I would say fairly well understood, limitation about how complexity is measured in statistics. Because what, in particular, the information criteria you're referring to do, and to some extent the MDL does as well, at least in its crudest form, is simply count the number of parameters. So that's what, in the AIC, that's all there is. It's the K, it's the number of parameters your model is estimating. And it is well understood that particularly when it comes to structural equation modeling, for example, that is not enough to really capture the properties of your model in particular how actually flexible it is, and so how likely you are to get a false positive, or in any case to be overfitting the data. So in fact, in another collaboration with a psychometrician in the US, in fact, Wes Bonifay, you mentioned it, at the University of Missouri, we're trying to see, I've been trying for a while with a series of pre-registered tests to see whether with these methods we can capture more of the properties of structural equation models than you would otherwise be able to do at the moment. So that is sort of a more technical, if you like, application, but because it's specific to statistics and models, but it's an extension that would stem exactly from this kind of approach. So the short answer is that I totally think that particularly the minimum description length principle is on mark as to getting at the bottom of what knowledge is supposed to be, but it's not going sort of deep enough and broad enough.